The purification of F1 ATPase is based on classical biochemical methods, which proved to be crucial for our understanding of the mechanism of ATP production by rotary ATP synthesis. The main advantage of this technique is that it produces a highly pure enzyme suitable for structure determination by X-ray crystallography or cryo-electron microscopy.
Although this protocol is optimized for the isolation of F1 ATPase from trypanosomes, it can be adapted to other sources such as tissue or culture cells and to various pathogenic protists. This technique may lead to the identification of new drugs to treat severe human diseases. For example, Mycobacterium tuberculosis F-ATPase is an authenticated drug target for the treatment of tuberculosis.
To begin the protocol, thaw and gently resuspend mitochondrial vesicles, also known as mitoplasts, previously isolated by hypotonic lysis, from 1 × 10⁻11 to 2 × 10⁻11 cells of procyclic Trypanosoma brucei, in 5 ml of ice-cold buffer A. Keep the sample chilled. Next, determine the protein concentration in the suspension by the bicinchoninic acid or BCA protein assay according to the manufacturer's instructions. Perform a BSA dilution series in ultra-pure water to construct the standard curve. Dilute a small amount of the sample 20 to 100 times with ultra-pure water to fit into the range of the BSA standards. After calculating the total protein amount in the sample, bring the protein concentration to 16 mg per milliliter by diluting it with additional buffer A. Then, fragment the mitoplasts into